Hello, this is part 1 recording about COVID-19. So in this recording, we will talk about the basics, transmission, evolution, the origins of the virus, effect on our organism, immune response, diagnostics, treatment, and the response from biotech and pharma industry. And actually, we will talk more about the different variants of the virus and vaccines in the second part of this lecture. So what is COVID-19? This deck of slides was made in the beginning of the pandemic. So at this point, we all know what this is. So it's a novel coronavirus infection caused by a virus called SARS-CoV-2. It causes viral pneumonia. It's related to SARS and MERS. It was identified in 2019 in Wuhan, China, and has spread globally. What are the symptoms of COVID-19? Again, we all are familiar with this information. It causes fever, body aches, dry cough, fatigue, chills, headache, sore throat, loss of appetite, and notably loss of smell. More severe symptoms include high fever, severe cough, and shortness of breath and pneumonia. Symptoms may appear anywhere between 2 to 14 days after exposure, and in some cases even later than that. In some cases, coronavirus infection may lead to the damage of the lungs, and imbalanced immune response may cause pneumonia. So in the beginning of the pandemic, there was this idea that the healthcare system couldn't meet the demands from those people with severe to moderate cases of COVID-19. So we were encouraged to stay at home and quarantine or limit our interactions with other people. And this is known as flatten the curve. So the idea was that by having people quarantined at home, it would lead to less cases and it would help to make sure that the healthcare system is not being overwhelmed. And here you can see healthcare workers from Buffalo, New York, wearing various forms of PPE. So as you all know, PPE stands for personal protective equipment and has become a part of the everyday vocabulary during pandemic. In the beginning, it wasn't clear if people should wear a mask and, should the mask only be worn by healthcare professionals. Some healthcare professionals were told that they do not have to wear a mask unless the person is coughing. Later on, this shifted to everyone should wear a mask. And nowadays, as the rates of COVID-19 infection are falling due to vaccination, many states have opened up and we don't have to wear a mask anymore. Though there are reports about a new variant called the Delta variant from India, and in Israel they have implemented mask wearing policy again, even though the whole country was allowed not to wear a mask. But now they went back to wearing masks because apparently the vaccine doesn't seem to be as protective against the Delta variant. So this hasn't happened in the United States yet, and we'll have to see how these policies will change. So how can we prevent infection with COVID-19? One way is to continue our good hygiene. Washing with soap and water can significantly slow the rate of the pandemic and limit the number of infections. So how does the science of soap works? It's actually quite interesting. So as you know, or you might know, our cell membrane is made of fatty acids. So it's a bilayer of fatty acids and phospholipids. So phospholipids are molecules that have a hydrophilic head and a fatty acid tail. So our cell membrane is a bilayer made of these phospholipids, where you have a layer of phospholipids on the outer membrane and the fatty acids are sandwiched inside, and then you have phospholipids again on the inner membrane. So it's a bilayer. And it turns out that the soap molecules are also made of this phospholipids with a hydrophilic head and a hydrophobic tail. So here we have a virus, a COVID-19 virus, with the spike proteins embedded in the cell membrane of the virus. This spike protein helps the virus enter the cell. And basically, what soap does is, because the viral membrane is also made of phospholipids, the soap molecules, which are similar in nature, they wedge themselves in and they pry the lipid membrane apart and destroy it. Another way that soap can help is it can form tiny bubbles called micelles, and they can, micelles, and they can trap viral antigens and residue from the virus, and basically then you can use water to wash the micelle away. So now let's talk about the origins of SARS-CoV-2. Where did it come from? So SARS-CoV-2, we think of it as a zoonotic disease that made a jump from an animal host to a human host. So the current understanding is that SARS and SARS-CoV-2 are beta coronaviruses that come from bats. And here we see this map here is showing you where the original SARS came from in China. And it was isolated from horseshoe bats, and they found that SARS sequences were similar to human genomic sequences. SARS genomic sequences in bats were similar to SARS genomic sequences found in humans. So what about SARS-CoV-2? SARS-CoV-2 supposedly originated in the Wuhan province of China, and one hypothesis is that it originated in bats, and then it jumped into an intermediate host called the pangolin, and then it infected humans. So one idea was that this happened at the wet market, where somehow these animals may have interacted. Recently, an international group of experts were in China trying to determine if this was true, and they came up with a new hypothesis that SARS-CoV-2 originated in some kind of frozen food that was transported to Wuhan. And of course, there is another hypothesis that the virus may have been engineered in the lab, and interestingly, there is a virology research institute located in Wuhan, China. The team of international experts who went to China most recently, they said that they have examined the labs and came to the conclusion that they did not have anything to do with this developing the virus in the lab. But recently, Trump administration and now Biden administration and Dr. Fauci have decided to look more closely into this theory of supposed lab origins of the virus. And so since, we talked about the fact that pangolin may have been an intermediate host, so what are pangolins? So pangolins are endangered species of mammals. They're very cute, as you can see. They have scales made from keratin, the substance that also forms human hair and nails, and they are highly sought on the black markets for their medical benefits. So those keratin scales are used in a lot of Asian Chinese remedies, so not just in China but in other Asian countries. And recently, Chinese government put a ban on using any pangolin scales in medicines. Pangolins can roll into a ball. They are easy to catch, and they're also valued for their meat, so the meat is considered delicacy. More than 1 million pangolins are believed to have been poached from the wild in the past decade. And yeah, so one good thing that came out of this pandemic is more protection for pangolins. So what are the origins of coronavirus? What scientific evidence do we have to say that it may have originated in bats and then jumped into pangolins? So what they have done is they compared the genomic sequences from SARS-CoV-2 that infected humans, from human hosts, versus SARS-CoV-2 sequences from bats versus SARS-CoV-2 sequences from pangolins. So if you look at this figure right here, we have this red dots representing human hosts from Nepal, Sweden, Wuhan, Japan, USA, and you can see that their sequences are very much alike, pretty much alike. And here we have a sequence from rat G. That's just the name of this bat sequence, and you can see that it's very similar to the human sequence, and in green you can see pangolin sequences, SARS-CoV-2 isolated from pangolin hosts. So this phylogenetic tree is showing you that pangolin has about 94 to 98% similarity in sequence to human sequence. Now, if you look at the SARS-CoV, which was the previous version of SARS, you can see that, it's believed that it was transmitted from bat to a feral cat called a civet, and then it jumped into humans. And here you can see bat sequences shown in blue, and civet sequences are shown in light turquoise, and then you have the human sequences shown in red. And you can see that SARS human sequences are quite a bit similar to civet sequences. Now let's look at, in figure B, let's look at the structure of SARS-CoV-2 genome and compare it to SARS-CoV and MERS-CoV, which are related, sort of the previous versions of this virus, which also caused severe respiratory symptoms. Notably, SARS-CoV-2 was causing no symptoms in China, and MERS was found in the Middle East. But ultimately, those two versions of the virus were not as infective and not as easily transmittable as SARS-CoV-2. So if you look at the genomic sequences of those three viruses, comparing those viruses, you can see that they all encode spike protein S, which is found on the outside of the virus and can help the virus enter the human cells. You can also see that it contains that 2/3 of the genome. So if you're looking at open reading frame 1A/B, encodes viral polyproteins, which form the viral replicase transcriptase complex that allow the virus to replicate its RNA genome. And the rest of the viral genome codes, like I said, for spike protein, shown in yellow. This domain is shown in yellow. Nucleocapsid protein domain is shown in red, and you can see that there's similarity between the species of the virus. And membrane M proteins are shown in green here. Okay. And interestingly, a rapid test for SARS-CoV-2 can be used to identify if a patient has nucleocapsid protein present in their sample, such as spit sample or like a nose swab sample. So this type of rapid test is similar to ELISA test, and it detects the presence of nucleocapsid protein. And we'll talk about the diagnostics of COVID-19 later on. So how does coronavirus enter inside the human body? So first, this is kind of a simplistic view. We will not get into the physiology of this, but the virus enters the respiratory tract. It could be through the nose or the throat or through the eyes. So basically, suppose if somebody is exposed to someone who is coughing, or somebody is talking, and you have the viral droplets, traveling through the air and landing on another person's surfaces, and that can allow the virus to enter the respiratory tract. It's interesting that in the beginning of the pandemic, it was believed that the virus is a large droplet virus, and it's not transmitted through the air, but instead it was believed that it's present on the surfaces. And there was this boom for cleaning companies. A lot of businesses hired cleaning companies to clean the surfaces. But now it's believed that it's a respiratory airborne virus that transmits through small droplets through the air. So it's airborne. And the virus enters the mucous membrane and starts replicating itself almost immediately. And it uses hosts' translational machinery to make viral proteins. The respiratory tract swells and is inflamed, and then the virus can enter the lung and the bloodstream, and more symptoms begin to show. So here we have the virus entering the human cell. So as I said, spike protein is instrumental. Spike protein is what gives SARS virus its characteristic corona-like appearance. So spike proteins binds to the ACE2 receptor found on human cells, and this complex gets internalized through endocytosis, and it enters the body. Another way is there's a sequences found on S1 protein which allow for human enzymes called furin enzymes, which are proteases, to make a cut on spike proteins, and this allows spike protein to enter the cell through another pathway without having to interact with ACE2 receptor. So this is ACE2 receptor independent pathway. So as I said, there are furin proteases present in the host, in the human, that cut spike protein in a specific way that allows it to interact with the human cell membrane and enter the cell. So once it's inside, it uses host translational machinery to make copies of viral polyproteins. The virus also has RNA-dependent RNA polymerase, which allows to replicate viral RNA. So viral proteins assemble with viral RNA, and they get packaged and create new variants, which can then escape the cell and infect more cells in the body. So now I'm going to talk about how the immune system responds to COVID-19 virus. This is a very complex figure from an early paper published out of China. And you can see, if you look at this diagram here, this part shows you how the virus enters the human cell, either through ACE2 receptor or through interaction with furin proteases. And here in this figure, we see the immune system responding to the virus. Here it is another more detailed figure. So this one shows you how the viral antigens are displayed for the immune system to notice them. And here we can see the responses from the immune cells. And finally, this shows you how the antibodies are produced by the immune system in response to the virus. So let's look first at antigen presentation, which is this diagram here. So when the virus enters the cells, it gets broken up into pieces called antigenic peptides. So these are just pieces of the virus. These are antigens, or foreign pieces of proteins, foreign proteins. And in the body, we have something called APC cells, or antigen-presenting cells. Those cells include macrophages, B cells, and dendrite cells. Think of APC cell as a waiter presenting the antigen on a plate. So it's APC cells contain special receptors that present the antigen to helper T cells and cytotoxic T lymphocytes, which are involved in eliminating cells infected with the antigen. So basically, think of APC as a waiter, and they're presenting you like a plate with the antigen on it. And so once antigens are presented, this activates a specific type of T cell called a helper T cell. So first, let's talk about the different types of T cells that we have in our immune system. So we have CD4 plus T cells that express CD4 receptors, and they are called helper T cells. So what is the role of helper T cells? They recognize the antigen presented by the dendritic cell. So you have this APC dendritic immune cell presenting the antigen, and it gets recognized by the helper T cell. And helper T cell releases special proteins called cytokines. And cytokines serve two purposes. First, they activate CD8 plus cells known as cytotoxic T lymphocytes. Additionally, cytokines from the helper T cells activate B cells. And then B cell can clone itself and make antibodies, which can be used later on to protect us from the virus, from another infection by the virus. So CD8 T cells can directly destroy infected cells. So once helper T cells activate CD8 plus T cells or cytotoxic T cells, you can think of them as killer T cells. So when cytotoxic T cell is activated, it interacts with an infected cell. So an infected cell will display antigen on its surface. Cytotoxic T cell will interact with this complex displaying the antigen. And cytotoxic T cell produces two types of molecules called granzymes and perforins. So what perforins do is they form pores in the plasma membrane of the infected cell. And granzymes enter the cell and break down proteins, and eventually they lyse the cell and it goes away. Okay, so it disintegrates. Okay, and so it turns out that one big problem in severe cases of COVID-19 is the immune system seems to create a very strong response known as cytokine storm. The report in Lancet journal, which is a very reputable medical journal, shows a phenomenon, talks about this phenomenon called acute respiratory distress syndrome, ARDS, which is the main cause of death for COVID-19. So one of the main mechanisms for ARDS is the cytokine storm, which is the uncontrolled systemic inflammatory response resulting in the release of large amounts of pro-inflammatory cytokines. So cytokine storm triggers a violent attack by the immune system to the body, causes ARDS, and this can lead to multiple organ failure and finally lead to death in severe cases of SARS-CoV-2 infection. Why does this ARDS response take place? It's not clear. Perhaps it's the body, many body systems are affected by COVID-19, and it's sort of like the last response trying to fight off the virus. So if somebody had COVID-19, can they get reinfected? do they produce neutralizing antibodies? So multiple studies at this point have shown that indeed they produce neutralizing antibodies and they can stay in the system for up to 2 to 3 months or longer. However, it's very difficult to control this and predict this for every single person. So at this point, they say that even if somebody had COVID-19, you can't be sure if they have neutralizing antibodies, so it's still recommended that they get vaccinated with COVID-19 vaccines. Here we have an image of one of the first patients who recovered from COVID-19, and this photo was taken back in March 2020. You can see here that all these medical professionals are not wearing masks because, like I said, this was in the beginning of the pandemic when there were no clear guidelines, and this is in Sri Lanka. This patient was actually on one of those cruises, on the cruise where they got infected with COVID-19. So how is this infection diagnosed? So one major method is RT-PCR. It's a very sensitive method that allows you to diagnose if somebody has COVID-19. Many of us are familiar with a nose swab, and then they do the RT-PCR. So the purpose of RT-PCR is to take the viral nucleic acid, which is RNA, and in the first step of this reaction, it's converted to cDNA, and then cDNA is amplified by PCR, and if somebody has SARS-CoV-2 virus, they would have the nucleic acid, which would get amplified by this method and detected. And if they don't have it, it wouldn't get amplified, and they would just have background values, and they wouldn't be positive for COVID-19. And ELISA is a way that allows you to detect the presence of viral proteins or antibodies against the virus. Point-of-care testing allows you to detect IgM, IgG antibodies developed in response to infection by SARS-CoV-2 or any coronavirus. So ELISA and antibody tests, serological tests, are not as specific as RT-PCR. It usually takes about 35 days for somebody to develop antibodies against COVID-19, so this is another reason why these serologic tests are not as effective. Also, doctors can use CT scans to determine if somebody has lung infection or fibrosis of the lungs, which is a dangerous condition in severe cases of COVID-19. So, RT-PCR can be performed rapidly using kits and specialized equipment such as CyFit kit. So basically, they collect person's sample, and they put it in a pouch, and then the pouch is inserted in the machine. And so this way, you don't have to do any additional pipetting. This can be done by a medical worker who is not trained in any molecular biology lab techniques. So they just put the pouch in. It already has all the reagents necessary to run RT-PCR, and then the machine produces printable results. Abbott is another rapid RT-PCR test that can be used, which was developed by a company called Abbott. At this point, many of you have been to drive-through testing, or, you can just come to a parking lot at CVS or Rite Aid or Walgreens pharmacies and get tested for COVID-19. Many colleges, like Bentley, have their own system for testing, and Bentley works with Broad Institute. And so what are some treatments for COVID-19? So a company called Celltrion is working on developing monoclonal antibody treatment for COVID-19. Companies like Regeneron and Eli Lilly have received an emergency use authorization for monoclonal antibodies. So what monoclonal antibodies do is they can be used at the early stages of infection, and they can prevent the virus from replicating inside the body. So monoclonal antibodies bind specifically to the viral protein, and allows the virus to be eliminated by the immune system before it has a chance to replicate. And vaccines, so again, this slide, like I said, is from the beginning of the pandemic, but now we have Pfizer and Moderna vaccines, which have received emergency use authorization approval. And I will talk about these vaccines in my second portion of this talk, but Pfizer and Moderna vaccines are known as RNA-based vaccines. And the way that they work is you have an mRNA, which is delivered via a lipid nanoparticle. And nanoparticles are made of phospholipids. And as you recall, I talked about phospholipids when I mentioned the science of soap. So phospholipids create this micelle, this lipid envelope, and essentially RNA is inside that envelope, and it can be delivered to the patient. Moderna also uses pseudouridine analog. As you know, the vaccine is made of mRNA, so instead of T's, instead of thymines, it has uridine, so U's. So the company modifies the uridine, which prevents APC dendritic cells of the immune system from detecting mRNA, which is introduced by the vaccine. So there's no immune response. And essentially the body becomes a factory for the production of S1 spike protein. The immune system learns to recognize spike protein as an antigen, activates helper T cells, which in turn activate cytotoxic T lymphocytes. And also helper T cells activate B cells, which can produce antibodies, neutralizing antibodies against COVID-19. And memory B cells are activated, which can help to produce antibodies in the future and prevent future infections by COVID-19. In the beginning of the pandemic, there was talk of using some old drugs as treatment, such as hydroxychloroquine, which became highly politicized. FDA revoked the emergency use authorization for this drug because it didn't prove to be very effective. However, some places, such as Henry Ford Medical Center, had some positive findings with it. And here we have some people who claim that they they were treated with it successfully. And here we have this actor here who actually is the voice of Benji in the new cartoon, Disney cartoon called Raya and the Dragon. So he had COVID and he said that he believed that hydroxychloroquine helped him get better. So the way that hydroxychloroquine works, it's an old anti-malarial medication, and one way that it might work is by preventing endocytosis, so preventing the virus from getting inside the cell. But again, at this point, it doesn't seem to be effective. It might only work for some patients with very early in the infection, in the course of the infection. Dexamethasone has been shown to be effective in patients with severe cases. Dexamethasone is a corticosteroid used as an anti-inflammatory, and it also has immunosuppressant effect. And it was shown to be especially effective to prevent ARDS and to prevent mortality in patients on ventilators. In the beginning of pandemic, there was also this drug remdesivir developed by Gilead company, and essentially it's an adenosine analog. So as you know, mRNA is made of nucleotides called A, U, C, and G, and adenosine analog is substituted instead of the A, and so it interferes with the RNA-dependent RNA polymerase and blocks viral RNA synthesis. And it was tested successfully with Ebola, and they thought that it might work for COVID-19, and it didn't really work that well, but it is often still prescribed to patients with moderate to severe cases of COVID-19. So now the protocol for treatment of patients with COVID-19 is the first line of defense is to give them monoclonal antibodies by Regeneron and Eli Lilly. So this is given through an infusion, through an IV. And the next thing would be if this doesn't help, is to treat them with like remdesivir or dexamethasone. And there are many vaccines that are being developed or that have already received emergency use authorization approval, notably Pfizer, Moderna, Johnson & Johnson, and Novavax is very close to receiving approval, emergency use authorization approval. Inovio in China is, I believe, has received approval in China. Sputnik 5 has received approval in Russia and is being used in different countries around the world. And like I said, Novavax from Emergent Biosolutions is a protein-based vaccine and is about to get emergency use authorization approval. So the question remains now, is it okay to use a combination of different vaccines like a DNA vaccine and a protein-based vaccine, or RNA vaccine and DNA vaccine? They have done some studies showing that it seems that that would be okay. It's not clear if this would lead to more adverse side effects. This remains to be seen. Here are some medicines. So these are not vaccines, these are medicines being developed for COVID-19. As you can see, Eli Lilly has developed monoclonal antibody. as I said, Regeneron also has a monoclonal antibody, and there are a number of other companies working on this. And you can see there are other ways, like they're trying to use small interfering RNAs, short interfering RNAs or RNAi, which we talked about in my course, and small molecules are being developed that can potentially inhibit SARS-CoV-2 virus.